Good morning. I'd like to thank the World Government Summit and the wonderful people of Dubai for welcoming us. I hope to convince you today to be as excited as I am about the microbiome, what I consider to be the frontier of medicine. And I want to start with a brief definition. The microbiome refers to all the microbes that live in and on the human body, not just bacteria, but also viruses, fungal organisms, parasites, protozoa, which are one-celled organisms, over 100 trillion in all. You can't see any of these microbes, but if you scrape them all up, they'd weigh about 3 to 4 pounds. We've known about the microbiome for a very long time. When Paolo Leonetti in the 1600s looked at his own dental plaque under the microscope. And I think the exact quotation was, he saw little animalcules so prettily a-moving, was how he described it. But it's literally taken us since then, about 400 years, to figure out that our microbes are actually friends, not foes, and that their existence is very much tied to our own health and well-being. Your microbes are actually a more unique identifier of you than your own DNA. They reflect everything about you, where you've lived, what you've eaten, the medications you've taken, whether you've had siblings, pets. A few statistics. We have about 100 trillion microbes in all, most of it in the gut. We have more than 1 billion microbes in just one drop of fluid in the colon alone. Our human genes are vastly outnumbered by our microbial genes. We have about 23,000 human genes and over 3 million microbial genes. And we have 10 times as many microbial cells as human cells, leading some to speculate that we are more microbe than we are human. If we think about what microbes do, I want you to imagine yourself as the beehive. Who animates us? Who actually does the work? Who digests the food and synthesizes the vitamins? Who metabolizes the drugs? Who neutralizes toxins? Who trains the immune system how to recognize friend versus foe? Who turns genes on and off? And who ultimately influences disease expression? It's the microbes. So the microbes are like the bees in the hive. So we, again, are really animated by our microbes in the sense that that is who is actually keeping us alive. This is a cover from Time magazine in 2010. This is a cover from Time magazine in 2010. Why your DNA is not your destiny. The thing that most excites me about the microbiome is a message of optimism. Because unlike your genes, as my 12-year-old says, you get what you get and you don't get upset, the microbes are constantly changing. Your microbiome that you walked into this room with is going to be different from the microbiome that you walk out of this room with, particularly if you take advantage of the wonderful fermented drink that our hosts have provided. There's some kefir in the back of the room, which is providing you with actual live microbes. So everything you do today, the food that you eat, whether you go outside and expose yourself to nature and some microbes, even the very thoughts that you think are all constantly changing your microbiome. So the possibility for change in terms of disease expression is vast. Our old paradigm of health and wellness said that our genes are essentially our destiny. Our DNA decides whether we get illness or not. We know from many studies, particularly studies looking at identical twins who have the same genetic material, but the expression of disease is not 100%, that there are many epigenetic factors that determine whether disease gets expressed. So genetics is a study of the DNA code that creates all the cells and tissues in our body. Epigenetics is a study of the things that affect those genes, the chemical tags that are added or subtracted to the DNA that ultimately determine whether a disease is expressed or not. So we have much more control over this than we suspected. And for most of us, illness is not something that falls out of the sky into our laps. It's something that, with enough research and thought, we can actually control. The Human Genome Project was the largest collaborative science project, started in the 80s and finished in 2003. 15 years, and it was an incredible undertaking of sequencing the entire human genome. And at the end of this extraordinary accomplishment, how many diseases do you think were cured by knowing the entire human genome? 0. Because the genes are just a suggestion. They are not what ultimately determine whether we get sick or not. New techniques in microbial research, the ability to rapidly identify the organisms growing in our body, to sequence them and divide them into communities, has dramatically changed our ability to research the microbiome. But even microbiome research, a human microbiome project, which was started in 2008, and looks at thousands of different samples, they estimate they've sequenced approximately 90% of the human genome. Even the Human Microbiome Project is not going to lead to the end of illness and disease, because we need to know more than just what these microbes are and even what they do. We need to understand the impact of how we live, our diet, lifestyle, environmental factors, the impact on the microbes. I want to play this for you. They're rolling up the green, and look at what's coming up. Smokestacks, in a minute, you'll see. Smokestacks coming up. You might recognize this video from the opening ceremony of the London Olympics in 2012. And this signifies the Second Industrial Revolution. And a funny thing happened when everyone moved from the farm to the factory. This happened. Autoimmune disease happened, what we consider our modern plagues. So in the turn of the century, in the last 50 years, we've seen a dramatic increase in autoimmune diseases, things like Crohn's disease, multiple sclerosis, asthma. I have an interesting story to tell you. At Georgetown University, where I work in Washington, D.C., we're very proud to train a lot of international gastroenterologists. We train many from the Middle East. And about 20 years ago, when I first arrived, we had a wonderful GI fellow named Hisham Salut, who was from Saudi Arabia. And I would invite Dr. Salut to come to my inflammatory bowel disease clinic, where I saw patients with Crohn's disease and ulcerative colitis. And he would say to me, Dr. Chutkhan, you're fun to hang out with, but the truth is, we really don't see these diseases in Saudi Arabia, so it's not good use of my time. Fast forward about 15 years, so 5 years ago, when I saw Dr. Salut at one of our GI conferences, and he said, we now have a clinic in Saudi Arabia devoted to Crohn's disease and ulcerative colitis. So what has happened? In the 1950s, David Strawn, who was a researcher at the London School of Tropical Medicine and Hygiene, was tasked with figuring out why they were seeing these skyrocketing rates, particularly two conditions in children in the UK, asthma and eczema, two autoimmune phenomena. And he embarked on a 30-year study. He looked at 17,000 children from birth to adulthood. And he made two startling discoveries. The first is that children from large families who were exposed to a lot of microbes because they were constantly being coughed on and sneezed on by their siblings who were bringing home measles and mumps and coughs and colds, those children had very low rates of autoimmune diseases like hay fever and eczema, asthma and eczema. The second, even more startling discovery was that children from affluent households where they had higher standards of personal hygiene, where they were bathing and sanitizing themselves all the time, they had the highest rates of these autoimmune diseases. Hashtag live dirty. This turned everything we thought we knew about medicine and health on its head. Could it be that it's actually better to be dirty? I will say I did clean up for you a little bit this morning. I did take a shower, but in general, I actually advocate less bathing and scrubbing and use of these products. So we are actually ruining our ecosystem. We are scrubbing away these essential microbes on our scalp, on our skin, and we're also ruining them internally, as I'll talk about in a moment. And this is inextricably linked to the increase in these diseases that we're seeing. So this shows a map. David Strawn's work formed the basis for what we now call the Hygiene Hypothesis, which basically says that in countries that are more developed, where the environment is more sanitized, and again, I'm not suggesting personal hygiene here, but more on a large scale level. So where there's large, widespread chlorination of the water, use of antibiotics in the food, in those countries, which tend to be in the more developed countries in sort of a northern distribution, we see very high rates of diseases. This map shows multiple sclerosis, but I could superimpose a map for distribution of Crohn's disease, of rheumatoid arthritis, of lupus, of type 1 diabetes. So very high levels of autoimmune disease in countries that are more developed, that have more aggressive hygiene practices or sanitation practices. What happens when we go from this to this? What happens when we take children out of the woods and off the beaches and we plant them in rooms playing Minecraft? Autoimmune disease happens, not an accident. It's not just what children are doing, it's also how they're born that dramatically influences a risk of disease. So here you see a table, babies born via C-section versus babies born in a more traditional manner. And we see higher rates of allergies, asthma, autoimmune disease, and obesity. And these changes actually follow children into early adulthood. This is a startling statistic. 1 in 4 babies in the US is born via C-section. And while some of these are medically necessary, many are done for reasons of convenience and commerce. People don't realize the effect that this single act of passing through the birth canal, this is when we are endowed with the majority of our microbes. We get them as we pass through the mother's birth canal. So having a baby born via C-section dramatically changes that baby's likelihood of having real disease. Similarly, babies who are nursed, the third most common ingredient in breast milk is something called an HMO, human milk oligosaccharides. Human milk oligosaccharides are completely indigestible by babies. Why is the third most common ingredient in breast milk something that babies cannot even digest? Because it's not there to feed the baby. It's there to feed the baby's bifidobacteria, the baby's burgeoning microbiome. Babies who are bottle-fed, again, much higher rates of these autoimmune diseases. So we have to think about these things, these medical advances that can be wonderful and life-saving, but also have significant consequences on the microbiome and subsequently on our health. Antibiotic use. India actually has the highest rate of antibiotic use, but the US has the highest per capita rate of antibiotic use. This is not a good thing to be number 1 in. Conservative estimates suggest that 50% of antibiotic use in the US is unnecessary. We're treating common colds and other minor infections that will be self-limited, and that also it's important for people to sort of suffer through from, because that trains your immune system. That's how you develop immunity. We're treating these with antibiotics, and the startling statistic, again, is that just 5 days of a broad-spectrum antibiotic of the kind that you might use to treat a sinus infection or a urinary tract infection will remove 1/3 of your gut bacteria. And those missing microbes are never coming back in quite the same robust way as when they were originally present. When people ask me about probiotics, I like to use the analogy. It's like taking a bathtub full of water and draining out all the water, and then taking a cup of water and pouring it in. That's the probiotic. And then saying, okay, I'm good. So I took 20 days of antibiotics for something I didn't really need to be treated for, and I took a probiotic and I ate a little yogurt, and I'm good. You're not good. Judicious use of antibiotics is essential, not just for preventing damage to your own microbiome, but preventing resistance to microbes worldwide and the emergence of superbugs. We know that antibiotic use is strongly linked to an increase in autoimmune diseases. It's also strongly linked to an increase in obesity. We've known for decades, since the 1950s, that when you feed farm animals antibiotics, you can fatten them up and increase in as much as 15 to 20% in their weight. 80% of all the antibiotics sold in the United States are used in the animal industry, in the food industry. And we are ingesting that. Food for thought. But we have a paradox with antibiotics. We know that antibiotics have revolutionized the practice of medicine, and the discovery of penicillin in 1928 certainly ranks amongst one of the greatest medical discoveries. Antibiotics save millions of lives every day. They could have prevented millions of deaths from things like the Great Plague. But we're in the age of Pharmageddon now. The pendulum is swinging the other way. Antibiotics cannot distinguish between beneficial bacteria that are essential for keeping us healthy and harmful pathogens. Antibiotics destroy everything, like a tsunami, destroy everything in their wake, not just destroying our microbiome, but again, leading to the emergence of superbugs. We know that there is an association between antibiotics and obesity and between changes in the microbiome and obesity. And this next slide I'm about to show you, I think, is one of the most incredible experiments done. This was research done by the scientists at Washington University in St. Louis. They took identical twins who were genetically identical, where one was slim and the other was obese, and they transplanted microbes from each twin into identical germ-free mice who had no microbes. So they took microbes from the lean twin and transplanted to one mouse, and from the obese twin and transplanted to another mouse, identical mice. And without any change in diet or exercise, you can imagine what happened. Microbes that were transplanted from the obese twin into the germ-free mouse, that mouse became obese without any change in diet. So we know that microbes can dramatically change things that affect weight. They can change the time it takes for the food to get from north to south in the digestive tract. They can change the secretion of hormones like insulin and ghrelin that are involved in deciding how much of the calories you ingest are stored as fat versus consumed. They can actually consume calories themselves. So the energy harvest, we used to think it's calorie in, calorie out. Not at all. And we've all seen examples of that, of people who eat all day long and never seem to gain a pound, and other people who look at food and it goes straight to the hips. So we know that it is not this calorie in, calorie out, but that our microbial complement, what microbes reside within us, have a lot to do with determining whether we are lean or obese. And unfortunately, we can't just borrow our friends' lean microbes, because at the end of the day, it's what you feed the microbes that determine who grows. In the last century, we've seen a dramatic decrease in communicable diseases, what we think of contagious diseases, like tuberculosis and hepatitis and measles. And at the same time, we've seen a dramatic increase in what we call NCDs, non-communicable diseases, the modern plagues I referred to: diabetes, heart disease, Alzheimer's, Parkinson's, etc., Crohn's, diabetes. The increase in these non-communicable diseases is linked to changes and damage to the microbiome. And what sorts of diseases are we talking about? Virtually every organ system. I don't have the brain on there, but diseases like Parkinson's and types of depression, schizophrenia, we now know it's not the only cause, but are linked to changes in the microbiome. Type 1 diabetes in the pancreas, inflammatory bowel disease in the gut, non-alcoholic fatty liver disease in the liver, an increasing problem, not just in people who are overweight, but also in lean people. Atherosclerotic heart disease, we now know, has a significant microbial component. Obesity, cancer, particularly colon cancer, pulmonary disease and allergies, and of course, rheumatoid arthritis and ankylosing spondylitis. So there are very few areas in medicine that we don't see a contribution for the microbiome. I'd like to tell you about 3 important studies looking at the impact of diet and the microbiome that I think you should know about. The first is a study by Paolo Leonetti and his colleagues in Florence, Italy. The Italians have really been at the forefront of microbial research, and much of what we know, we owe a great deal of gratitude to them. Paolo Leonetti set out to study 2 groups of infants. Both were born traditionally and both were nursed. Infants in Florence, Italy, and infants in Bulpon, Burkina Faso. And at birth and through early toddlerhood, these infants were the same in terms of their microbiome. So again, they were born traditionally, not via C-section, and they were nursed. But as soon as the infants graduated to table food, to the local diet, everything changed. Well, what were the kids in Florence, Italy eating? They were eating actually a standard American diet, which is very similar to the standard Italian diet. They were eating pasta and pizza and gelato and ossobuco. So a high-fat, high-animal protein, low-fiber diet. What were the kids in Burkina Faso eating? They were eating root vegetables and lentils, whole grains, mostly things that were grown locally, and I believe the article quoted, enlivened by the occasional termite or free-range chicken for animal protein. So they were eating a plant-based diet that was unprocessed. And what happened when the kids started eating these different diets? The kids in Italy had a dramatic increase in microbes that were associated with diarrheal disease, with obesity. They had low levels of something called short-chain fatty acids, which are products that are produced when bacteria ferment poorly digestible fiber. And short-chain fatty acids are a marker of good health in the gut. The higher the level of short-chain fatty acids, the healthier the gut. They're used as an energy source by cells in the colon. The kids from Burkina Faso had microbes that were associated with leanness, and they had very low levels of these microbes that were associated with diarrheal disease. They had double the levels of short-chain fatty acids. Now, what's important to notice is that neither group of children were actually sick. Both groups of children were well, but the 2-year-olds in Italy who were eating this diet had microbes that were predicting disease to come. And the African children in Burkina Faso had microbes that were predicting health. Again, their main concern is communicable diseases, not these non-communicable diseases. Now, you might say to me, that's a very interesting study, but there are vast environmental differences between Florence, Italy, and Beaupont, Burkina Faso. How do you know that it wasn't those environmental differences that led to these dramatic changes in the microbiome of the children? So let me tell you about another study. This was a study done at Harvard a few years ago. They took 9 volunteers and they fed them initially a high-fat, high animal protein diet. It was essentially an Atkins diet, for those of you who are familiar with it. It was prosciutto and pork rinds and cheese and just all animal protein. They put them on that diet for a couple weeks, and then they rested them for about 5 days. And then they took the same volunteers, and they put them on a plant-based diet of lentils and jasmine rice and tomatoes and fruit as a snack. And then they looked at the microbiome, and they looked at the microbiome with each of the 2 diets, the same group of people, before, during, and after. And what they found was that within about 30 hours, so less than 2 days of the food hitting the gut, everything changed. The bibliophilia, the bacteria that are bile-loving, that are associated with breaking down animal protein and fat into its constituent parts, but are also unfortunately associated with inflammation, increased dramatically. So the bacteria changed within about 30 hours, but even more startling, the genes that were turned on and off also started to change. So again, this is a message of hope and optimism, that by changing our diet, something as simple as what we put in our mouth and put on our plate, we can actually influence the likelihood of whether we develop these diseases or not. Now, we can't influence it all. There is sometimes something called bad luck. But this idea that your genes are your destiny, not the case. So this is the second study. The third study was a very clever study done between the Imperial College in London and University of Pittsburgh, where they took 20 African Americans from an urban setting and 20 rural Black South Africans from a rural setting. And they swapped the diet under very closely controlled circumstances, so they knew exactly what they were eating. They swapped the diet for 2 weeks. And as you can imagine, the American group on the African diet fared very well. They had less inflammation in the colon, and they had reduced markers for risk for colon cancer. But the poor South Africans, they didn't know what hit them. They had a dramatic increase in multiple markers that confer an increased risk of colon cancer within just 2 weeks. So what were the Africans eating that was conferring such a health benefit? They were eating MACs, microbiota accessible carbohydrates. Lentils and other legumes, millet, rye, whole grains, root vegetables, indigestible plant fiber. And when you eat a lot of indigestible plant fiber, so we now know from the Burkina Faso study, we know from the Harvard study, we see here from the Imperial College Pittsburgh study, indigestible plant fiber. And we know when we eat a lot of this indigestible plant fiber, which contains something called inulin, that we grow a lot of this bacteria, Faecalibacterium prosnitzii. I really just like saying the names of these organisms. We shorten it to F. prosnitzii, but Faecalibacterium prosnitzii, it's just so wonderful. And it is a wonderful microbe, because this microbe can protect you against cardiovascular disease, colon cancer, diabetes, and obesity, these non-communicable diseases. And vegans, real vegans who actually eat vegetables, not the Pop-Tart and cereal vegans, vegans as well as what we call flexitarians, people who eat some animal protein but eat a lot of vegetables, have high levels of F-prosnitzi, and the higher the levels of F-prosnitzi, the higher the levels of short-chain fatty acids, the healthier the microbiome in the gut. This is one of my favorite quotations from Proust. The real voyage of discovery lies not in seeking new horizons, but in having new eyes. We have to have new eyes when we think about health and wellness. We have to think about our inner terrain, not just externally what's going on in the environment, but what's going on in our internal environment. And this is a quotation from Michelle Williams, who's a dean of the Harvard Chan School of Business. Uncovering how the human microbiome interacts with our organ systems stands to revolutionize how we approach human health, disease prevention and detection, and potential treatments. So let me just spend the last few minutes talking about some of these ways that we may be able to revolutionize our healthcare system. The first is really addressing overuse of antibiotics. If we have a healthy microbiome, if you can improve the individual microbiome, we have less need for antibiotics because illness and infection specifically is generally an issue of terrain, your internal soil. Pasteur's germ theory tells us that we get sick because one bad actor, a bad microbe, gets into our body and makes us sick. And that's true in some cases. Ebola, bad actor, it's going to make a lot of people sick. But most of the things that we're suffering from, when we think of respiratory tract infections and sinus infections and urinary tract infections and even skin infections, have to do with our internal terrain being unhealthy. We maybe had the misfortune of being born via C-section. We were bottle-fed rather than nursed. We were picky eaters. We ate no vegetables. We lived inside and played on the computer all day. We had no pets. We didn't get exposed to microbes. Perhaps we didn't have siblings to cough on us and make us sick, and now we know make us healthy. And then we started getting medicated. We started taking antibiotics. We perhaps were over-vaccinated. We started taking acid-suppressing drugs that decrease stomach acid and allow pathogenic bacteria to overgrow. We started taking hormone replacement therapy, steroids, all these different things that dramatically affect the microbiome. And lo and behold, now we get sick all the time. And we wonder why. So really looking at our terrain and how healthy our gut garden is can really help us to determine how we get sick and why we get sick and how we can get healthy. So again, by improving the immune system and supplying the microbiome with protective species, not so much through taking a probiotic, but by being more careful with the medications we take, our diet and lifestyle, so that we can safeguard our existing microbes. If we look at antibiotic resistance, which is really a global crisis, how can we improve our knowledge of antibiotic resistance? When we do that, we can save lives, we can prevent hospitalizations, we can decrease the death rate, and we can curb healthcare costs. So this has broad implications on a larger scale in terms of what governments can do. What about malnutrition? We know that 2 billion people suffer from malnutrition, and we see something that we call quality starvation in the more developed world, where people have plenty to eat, but no actual food. Edible food-like substances, high caloric intake, low food quality, a different kind of malnutrition, and perhaps even a bigger threat than just not having access to food. We know that malnutrition costs us 5% of the world's GDP, and that there are socioeconomic obstacles to dietary change. But perhaps if we can't change the food, we can change the gut, and we can make it more effective at producing those short-chain fatty acids that are so tied to our health. We can develop new strains of bacteria that can perhaps mitigate the toxic effects of food, that can enhance nutrient absorption, that can neutralize carcinogens and improve nutritional status. And I believe our next speaker is going to have a very exciting talk that incorporates a lot of these aspects. So we really think about decreasing non-communicable diseases. We know that currently 70% of global deaths are due to non-communicable diseases. And so as we think about how we approach things like diabetes and Alzheimer's, cancer and heart disease, we really have to think about the root cause of some of these diseases and our internal terrain and how our internal terrain affects the likelihood that we will develop one of these diseases. I want to leave you with a short video that illustrates what's happening in the external world around us. We are in the largest species die-off since the dinosaurs. In the last century alone, 80% of the world's forests, 70% of the fish, and 50% of the wild animals on the planet have disappeared. We're losing species at a rate thousands of times faster than what should occur in the natural world. And that's not just externally. Internally, within our microbiome, we have only 2/3 of the species, the microbial species, that our brothers and sisters in the Amazon have. Rewilding in conservation ecology refers to reintroducing species to improve the health of the external terrain. We have to think about how we can rewild our cells, our bodies, our skin, so that we can regain our health. Thank you so much for your attention.